Thank you so much for the amazing outline of the policies and the pillars for open science. And so I guess my job here is to tell you a small story about implementing against those policies and trying to actually deliver on, I think, the vision. Before I start, I thought I'd tell you one anecdote and one source of immense anxiety. So the anecdote, I think, is from 2013, 2014. Just about ready to head off to holiday in Greece. And I get an email from Antje saying, I want to write a proposal. It's around this. I want a global network. I want to do this. And I want you to do the following. And I want you to write this. I want you to write that, and so on. And, okay. So I spent my days on the beach, nights at the computer. And every night there was, okay, do this, do this, do this. And that was the Global Bioimaging Grant. And look what happened. So the learning from there is when Antje tells you to do something, do it, okay? Follow directions and follow the coordination. The source of anxiety is somewhere very, very late in the evening. I got an email from Yara saying, Jason, here's how I want you to lay out your talk, okay? I want you to start out here, make these connections. I really want you to finish in this form. I really think that's a good idea. I think you should do that. I think that was more or less what you wrote. And so I'm up against it here. And so with that, I'm going to start moving. I'm going to tell you about a story that we've been working on for some time. But that story is informed by the problem that we've been talking about for the last few days, and I think that governs a lot of what we do in this imaging data world. We have the privilege of working with great pictures. We make great presentations, but obviously we want to make the measurements that are our science. Over the last many years, we had this aspiration, and now we have the reality of a public resource. And I think for the last, recently, we're differentiating between data that's public and data that's AI ready. And so that's just starting, and so that's the next step. So that's what draws us together, and the question is, how do we solve this problem? I got into this business as a graduate student. I stumbled into, completely by accident, the first laboratory in the world to put a CCD camera on a microscope. In 1989, 1990, notice no GFP here. We're purifying proteins and labeling with fluorophores. We were injecting proteins into cells. This was Drosophila embryo and taking time-lapse movies. It was the craziest thing that anybody had ever seen way back then. And then, armed with about 10 time-lapse sequences from 7 years of a PhD, I finally graduated and moved on. By the time I started my lab in Dundee and started taking forward time-lapse imaging in various ways, the systems were commercially available, and now we're taking hundreds of data sets. And so the question is, how do you handle that data? And we were in the environment, you could see, I was trained at UCSF, worked with a lot of structural biologists. The PDB was coming online. You could see how important public data was at that time, and the first genomes were starting to become available. And that raised the question whether the data sets that we were collecting at Dundee and others were starting to collect in all sorts of different biological systems, et cetera, could come together in some way, be connected, and without really defining what that was, because we had no idea what we were talking about at that time, and be available for collaboration, for example, from my lab to yours, ultimately as a public resource, maybe as something like the PDB. At that time, there was a big effort to annotate genomes. So could we get a way to properly structure and annotate imaging data so that we had a common way of representing it? And oh, by the way, in the early days of the web, all of this would be available. And so those were all questions. Could we do any of those things? And it's amazing to be able to stand here with all of you, and so many people in the room have contributed to this, and say, actually, yes, we can do this. We can do this for real. We're still on a journey, as we've heard over the last couple of days, but this is coming together. So what I'm going to tell you about is our contributions in Dundee from a project I founded in about 2001 called OME, so the Open Microscopy Environment. And this is really all you need to know. OME is an absolutely committed to an open source model, and we're building image informatics tools. So what does that mean? What is informatics? It's really the tooling that makes communication possible in data and in analytics. That does mean that we don't do a lot of the stuff that gets Nobel Prizes. You saw that in Jan's talk earlier this week. This is incredible work that's happened over the last several decades, but others do that. But that data gets generated and then has to go somewhere, and it's that go somewhere that we work on. We connect into processing tools. We don't do the great new analytics, the new AI, but we try to enable that. We make sure that there's data is under management, controlled, access, et cetera, tagged, and so on. Bolting on many different analytics. I'll show you a little bit of that in a second, and hopefully as well visualization. So the stuff in the middle, the stuff that connects to these different functions on the periphery of the slide, that's what we're all about. That stuff is software. Okay? It's open source software. The people who are more technically minded in the world, in this room, know that that's APIs, effectively interfaces for connecting between each other. Okay, so OME releases the open source software, many different tooling, and you've heard many of these mentioned in the meeting. I'm going to give you a quick introduction to bioformats. OMERO, spend a little bit of time on IDR, finish up with a little bit on the NGFF work as well. These tools are used all over the world in all sorts of ways. So we started off as bona fide molecular cell biologists, still are. But this is, as I was telling Vlad, this is used for pollen counting in Panama. This is used in food manufacturing in large corporations all over the world and many other places. It's been great to see the pickup and all of the opportunities that people have taken with the work that we've delivered. Okay, so just to sort of introduce you to how we work. So we imagine, and I think this is probably true listening to everybody over the last couple of days, imaging facilities all over the world, stacked with various types of different imaging technologies, and the data rates here are rough. Probably some of you will say, well, they're actually an underestimate, but they're significant. But the key point is that these systems largely are commercial platforms. They produce data in a variety of different file formats. Those will be deposited on some sort of institutional file server. And bioformats is a file format translation system. So it looks at files, identifies them, and on the fly, and that's a really important concept I'm going to keep coming back to, on the fly converts the data, the metadata, and the binary data in these native file formats to a common model that something else can use. And bioformats is a massive success story for us. Started hundreds of thousands of times all over the world every day. Many people use bioformats for many different things. We take bioformats and put OMERO on top of it. And inside one little sort of ellipse here is a large data management system and interfacing, et cetera. And the key point is that what this tool does is make access to all of this possible through a secure connection through the internet. So just like you buy your airline tickets or you talk to your bank account, the idea is that you can do the same thing with this type of data with OMERO. That's sort of the tagline I use for non-scientific audiences. So, a web browser up here can see all of this without actually having to engage with it. And equivalently, analytics, clusters, et cetera, all can work. Again, we do this part, and then we bolt on to other tools up here and other tools that, here that are built by others. So hopefully that's clear. The next couple slides are going to be screenshots of a web browser that's talking to OMERO, talking to bioformats, and talking to all of this. So what you're going to have to imagine is in the next few screenshots, I'm going to show you this, but everything underneath is there. So the first one are DICOM images, and so these are obviously brain MRI. Many of you will recognize the metadata over here on the right-hand side. These are DICOM tags. This is the standard layout in our web client that we call OMERO Web, and so file organization, thumbnailings, and metadata. But remember, bioformats is sitting underneath this, so we can point that same application to, this turns out to be high-content screening data, and this metadata over here is results from Cell Profiler, so we can integrate all of that. And one more quick example, this is TCGA, the Cancer Genome Atlas that Steve mentioned yesterday. So this is whole slide imaging data, I think off of an Apereo system, and the metadata coming off of the TCGA analysis. And so what you're seeing here are that these tools being able to adapt to different metadata structures, but present them in a common way, in this case, to a web browser. But all of this is also accessible from from an API that can be accessed from many different languages from the command line as well. Okay. So that's what we've been doing for the last 20 odd years. And that's worked pretty well. I'll set one slide on analytics. So, I've sort of said that you can connect this to many different analytic tools. Here's an example of the tooling that we know that we've used in various applications, et cetera, that connects to OMERO, some commercial tools, many open source tools, et cetera. And so this common interface or this common API provides access to scientific tools that ultimately deliver the discovery. So we're doing the infrastructure, and these tools provide the way to get the analysis done to ask the questions that are so important in science. We've sort of, over the last several years, have a small sort of side industry in training workshops. So this is just one screenshot. This was at CRG Barcelona, I think at the end of last year. Our teams being invited all over the world to provide training, certainly on OMERO, but I think more importantly on what does data management mean, best practices, et cetera, and we're very happy to do that. So if you want to see where those are, just search for open microscopy events. We list them. And if you have any questions about that, let me know. Okay, so we had these tools. And so I told you it's a story that goes up to circa around 2014, 2015. And it became clear that this idea of publishing bioimaging data as a public resource is something we should need to think about. And we also might have the technology to be able to do that. So we started having this conversation with our funders about building a globally accessible, public, multi-domain bioimage data repository. And this idea of bringing these bioimaging data sets into, converting them into a reference community resource. I want to emphasize this is not just us. This was in the context of Eurobioimaging in particular and Elixir. But our role was to think about the technology that would enable this vision. And so we started down that road. Surprise, surprise, it's OMERO again. But the idea, at least, would be to take many different types of data sets, integrate them in some way, have all of the annotations, so a lot of curation here to make them as valuable as possible. You heard Shuichi and Matthew talk about this idea of additional added value metadata. That was definitely our aspiration. Make all of this available in a web browser, analytics, download, et cetera, and make all of this happen. I can confidently say, this idea was something we were pitching sort of from around, I would say, 2013 to 2015. We're very grateful in the UK for the BBSRC for funding this in collaboration with our colleagues at EBI. This is now up and running at their collaborative embassy resource. And I just want to say this openly, they don't charge us for something that we estimate that it costs about $250,000 a month on Amazon. So that gives you a sense of the scale that we're working on and how important that collaboration is. Just a few stats, lots of data, lots of files, lots of organisms, lots of annotations. That's that high added value. And lots of usage. So about 100,000 unique IPs in the last year, about closing in on 200 million hits. A quick estimate from Joe's slide, it's a couple % of EBI, which doesn't sound, for me, I think that's pretty good. But yeah, so it is really a global reference resource. And the idea is that published papers can connect to data sets that are published online. There's DOIs, et cetera. And there's, in the data availability section, there's a reference to the resource. Quick tour of a couple data sets. This is the reference embryonic fate map of the mouse embryo from Kate McDowell and Philip Keller. A few others, data from the Human Protein Atlas, the Tissue Atlas. They have this amazing resource, but the raw data weren't available. We've published about 12,000 antibodies worth of their data on IDR, and continue to work with Matthew's team at the BioImage archive to take that forward. Lots of annotations, sorry, lots of annotations. Never would we come up with our own controlled vocabulary, so everything is reusing things that we can find that are available. That's all queryable. And recently have been getting into this idea of multimodal data. And just because of the discussions, I wanted to show this example. This is a data set, obviously not ours. This is from Fei Chen at Yale. This is in situ sequencing. So we have the images, obviously. We have oops, I'm getting mixed up here. We have the regions. And in every one of those spots, effectively, we have the UMIs and all of the metadata. So sitting under OMERO is a tabular store where we can start putting that genomic data, et cetera, in there as well, and that's all working. Others have then picked that up, not us, and then made applications on top of it. This is Jean Ma's lab at CMU, so it's a nice example of data reuse published in Nature Methods. We were sort of naive to all of this effectively, but they've made a nucleome browser out of this data set and others that we published. Okay. So on one slide, I think we've published now about 140 data sets. And so we've been running this since 2016, 140 data sets. I mean, go back to Joe's presentation, that's not very much. You know, what's the difference here? And the difference is we're focusing on this idea of these reference images, these high value, comprehensive studies across, for example, genes, compounds, or other biologic systems across many different domains. And it's that reference characteristic that's so important to IDR data. That raises the question, obviously, of what about everything else? And I think you already heard the answer beautifully from Shuichi and Matthew. This idea that we published with Jan and his colleagues at EMBL in 2018, this sort of differentiation between repositories and added value resources. We've been following that pattern, and it's been very productive. And as you see, now we actually have a global ecosystem going forward. That's very exciting. So if you want to submit data to IDR, there's the email. The question is, you know, where to go from here? And circa, Josh, about 2017 or 2018, this new technology had been around for some time, but it started to become relevant for bioimaging. People were talking about this thing. There is a definition that a cloud is just somebody else's computer, and there is a bit of that. But actually, it's more than that. And in particular for bioimaging, the technology that sits under these cloud architectures for storing data is very, very different than what we had been using. And so that forced a reimagining of how we write down data, how we use data, how we access data. And Josh told you about this whole idea of NGFF. So I'm not gonna repeat any of that. I'm gonna provide a scientific rationale and examples of why that's so important. So why should we do this? Is it just to get data to somewhere else? If that's really the only driver, then that's actually not very interesting. It does provide a streaming access, so think of Netflix, et cetera, but now for bioimaging data, and you saw examples of that sort of hyper zooming in and out. Okay, so that's all great. So visualization is nice. That's basically Netflix for bioimaging data. It makes that possible. Federation is probably one of the most important things, I think of all of the data sets that we've seen. How are we going to take all of those forward in the future? And ultimately, this integration of many different modalities. And I think that's really where these opportunities come to the fore. So I'll give you a couple examples of this. I'm not going to show you, you've seen this sort of chunked idea of data at different resolutions. That's been shown by several speakers, so I won't reiterate that, but that's all working in this OME-ZAR format. What's probably more important, and that's hard if you have to grab, the idea is that's hard if you have to grab 1 terabyte of data. It's easier if you can grab 1 megabyte of data. So that's roughly the idea. Key point is that Josh pointed out really nicely is that metadata, this ZAR format provides us a way to start standardizing and sharing metadata. We've talked about that over and over again, how important that is, the REMBI specification, et cetera. But ZAR provides a way to represent that in a file structure that's easily accessed in parallel, so we can access that from a URL. And so, this is just a bit of an example of it. but that specification is open. There's an active community that's building different parts of it. It's extensible, human and machine readable, follows web standards, so we can turn that into URLs. And we actually now have software that converts proprietary file formats into OMEZAR. That's called Bioformats to Raw. That's from Glencoe Software. Have a look at that. So the pieces of this are coming together. These metadata structures are now getting standardized. We have a lot of work to do, and acknowledge an enormous amount of work from Josh guiding the community towards specifications and moving through the various versions. And I just want to re-show you this because we're very, very proud of this. We think, and I'm probably out on the edge here, so happy to be slapped down, but I think this is the world's first federated bioimage data resource. It's just a stack of CSVs. I think I have 4 grants that have gone down simply because I proposed that a federated bioimage data resource was necessary, and the reviewer said, "You're crazy, you'll never do that. It's impossible, no one will use it." There you go. So why is that so important? And here I want to take a bit of a diversion. Why are these data sets going to be so important in the future? And so some of you know that for the last 3 years, and Antje mentioned this, I've been running a program called Delta Tissue at Wellcome Leap. And the idea was to look at tissue state transitions. And the rationale for that, why did we want to do that? Because tissue states, changes in tissue states, are changes in disease states. So as a tissue state develops that's somehow pathological, that's a disease. And as that state evolves or goes through transitions, that's the disease progression. When we talk to clinicians, they said, I'm pretty good, emphasis pretty good, on diagnosis, on measuring tissue state. But what I can't do is predict tissue state transition on anything like the time scale that's meaningful for patients. And so we were struck by that idea, knowing that imaging was a key to start looking at tissue states and could provide the data to predict tissue state transitions. We looked at 3 different diseases as sort of demonstrators for a whole bunch of technology that we developed. The website is here. The program ran for 3 years. It finished on the 30th of September, which means I'm getting a little bit more sleep at night, which is great. The reason that I wanted to kind of introduce this to you this is the justification, for me at least, for why we're focusing on OMEZAR and integration. When we were setting up the program and looking at the range of different, how we would look at tissue volume versus tissue spatial resolution, so that's the horizontal and the vertical axis. Note the different colors of the different species we might measure and the different markers. When we looked, we thought about what we would want to do for example, profiling a heterogeneous tumor or heterogeneous TB granuloma. We knew that these were very large volumes, and we knew that we were very good at, for example, looking at something that was orders of magnitude smaller. And we knew that our spatial resolution could take us from tissues down to single molecules, but we knew also that if we wanted to properly profile a tumor, for example, at molecular resolution, we want to be in this sort of what we call this target zone. And then when we started looking at the different modalities that were available, and these are just some of them, I've stripped a lot off the slide, they're great, but nothing was anywhere near. We were orders of magnitude off of where we needed to be. So we can't just use my favorite instrument. We actually need some kind of data-driven, objective way to select which instruments we use for the different diseases. We're going to have to integrate the data from those to somehow get a representation that gets to this region. Okay? And oh, by the way, we're going to have to do a hell of a lot better with the technology. So we did all of that in the program, and you're going to start seeing papers on this. Gordon Mills showed you work from the Durban team on human tissue TB platforms. We know now that there's 4 different types of triple-negative breast cancer that have different therapeutic targets that we're working on, going into trials, and we have a path to trials from glioblastoma. 3 companies started, 2 more in the works. All of that in 3 years. It's been an amazing ride, but the key to this was multi-dimensional, multi-scale, multimodal data. Okay? So my statement is that this will, so all of the work that we've seen over the last few days are the key, will be able to drive the insights into the diseases that we're all studying. This will not just be, our facilities will not be convenient places to ask nice questions, get nice pictures. They will be places where discovery and therapeutics are derived on the timescale of a couple years. Okay? That's a big change that's coming. These data sets allow that. Okay? This is an example, H&E image of a tonsil tissue. This is fluorescence and segmentation masks. Behind the segmentation masks are feature vectors. These are actually transcript profiles. All of this data in a single ZAR. All of the things that Josh was showing you now in this multimodal representation. Okay? Josh emphasized the community. I'll just say that the authors on our papers span many different institutions, and that's great. It's an open conversation. Please join us. So just to finish up, I've shown you lots of technologies. All of this is available going forward. I think the NGFF strategy enables the directions that we need to go forward. We now have about 4.5 years of funding from various groups, certainly from the Dundee group, Jerby, Shuichi's group, Matthew's group, et cetera, others, I'm sure. So hopefully you'll be seeing a lot more of this technology coming available. I would say, just to finish up, this combination of these many different, high-level parts of the community all coming together. This is something that when Antje wrote me was just nascent, just coming together and saying, I want to do this. But now we actually can look around and say, this is all for real, and that's a huge accomplishment. But I would come back to what we ultimately have to do. We have to change people's lives. And I have to say, I've just been so much enjoyed seeing our software being taken in a direction that I never imagined. One day in my email, I got this picture. Medical students in Indonesia. You saw this picture from Ari. Medical students from Indonesia. So the first thing I saw is like, that's OMERO. Okay, that's good. Second, lots of medical students, and we saw them logging into our server. So it's like, now I understand where they're coming from. Lots of women. Interesting. So the training programs in Indonesia are really driving women, and Ari, something like 2/3 of the women are medical students. Yeah. Interesting. Thinking about the UN Sustainable Development Goals, a lot of this is aligning right there. And I would just say, and I really want to emphasize, we provide this open source technology and really ambitious people. And here we have to acknowledge Dewa, who is this incredible graduate student who's now in Kobe. Shuichi, you should grab him before he goes back. Built all of this with our stuff, never asked us anything, and built this whole system. And only when it was about to break our servers came to us and, did we discover, oh, there's something here. We need to actually start collaborating. I think it's an amazing story. So thank you all. Thank you to the team that's done all of the work. Thank you to the funders. Collaborators in particular, I would highlight at EMBL, EBI, Alvis Brasma, Ojas Sarkans, and now Matthew Hartley, Shuichi here in Japan. Collaborators in the US, and in particular, Ari. It's been an amazing ride to see everything that's been going on. I will stop there. I'm 2 minutes over time, and so I'm going to get kicked. Thank you so much. And